Hello everyone. So the topic of my research is the bioenhancing effect of curcumin and piperine with tamoxifen on cell viability of A375 melanoma cells. So I'm going to start with the introduction. So melanomas are often formed by melanocytes, which are skin pigment producing cells, and it is one of the most severe form of the cancer cells, and where most of the techniques and lots of strategies are being used nowadays to increase the effectiveness of the existing anti-cancer drugs, such as using the bioenhancers to increase the efficacy of anti-cancer drugs. So the hypothesis of this experiment is to see the bioenhancing effect of bioenhancers such as curcumin and piperine of tamoxifen using A375 cell line, which is melanoma cell line, and it was achieved using by extracting curcumin and piperine using SFE and then using UPLC analysis, and then by MTT assay for to cell viability and scratch assay for their metastatic effect of the cells. Moving forward to methods, so first of all we extracted curcumin and piperine using supercritical fluid extraction, and it was extracted from turmeric root and black pepper using CO2 as a main solvent and ethanol as a co-solvent, and then the extracted product was rotary evaporated and resuspended in methanol, and then SP analysis was performed, which was solid phase extraction, and then pre and post SP was performed and the recovery rate was found, and then using UPLC analysis and standard curve, the identification and quantification was carried out of both the standards. Then cell culture, cells were cultured in DMEM media, which is Dubesco modified eagle medium using 10% FBS and 1% pen strap, and it was grown under the condition of 37 degrees Celsius with 5% CO2. And the figure shows the 70% conference of A375 cell line. Once the cells were grown, then they were seeded at the density of 1 to the 10 to the power cells per mil for the MTT assay, and after 24 hours the cells were treated using the following treatments. And there were two separate treatments, one treatment of curcumin piperine with tamoxifen and the other treatment was without tamoxifen, and the result for both of them was compared to see the respective effect, and the cell viability was measured using biotic citation 5 and after performing MTT assay. The other method performed was a scratch assay. So once the cells were raised to the desired density, the monolayer of the cells was scratched using the pipette, and the respective treatments were added in each valves. And in this case, there was a no tamoxifen in each of the valves, and the result was imaged after 24 hours. And the quantitative analysis was not performed in this case, but only qualitative analysis was performed, and the statistical analysis was carried out using one-way ANOVA, and it was expressed using mean plus or minus standard deviation. Moving forward to the results, the UPLC results showed us that the retention time of the curcumin was 9.2 minutes, and the piperine was 9.3 minutes. The samples were identified using the respective standards. The recovery rate after performing pre and post SP were for curcumin 90% and for piperine it was 85%. Whether in case of cell viability results shows that as shown in the figure A, curcumin showed less cell viability whereas piperine showed the protective effect against the cells. Or else it can be said that piperine showed protective effect, so it was growing more cells, whereas curcumin was killing the cells. And then figure B shows the curcumin effect with combination of tamoxifen, and it showed the decrease in cell viability, but though it did not affected the efficacy of tamoxifen, whereas in figure C the piperine was combined with the tamoxifen and it didn't show the decrease in cell viability, but it did show the increase in cell viability. That means the cells were growing even after using the piperine. That told us that piperine was giving the protective effects against tamoxifen as well. And the last was the cell viability results after performing the scratch assay, it shown that curcumin and piperine alone without the tamoxifen, which shows that the cells on the scratch of piperine appear less compared to the negative control sketch, and the curcumin treatment compared to the scratch of the negative control will also appear low. So based on this, the non-metastatic behavior of cells was observed in the presence of curcumin and piperine on cells. Moving forward to discussion, uh, it was of cell viability and metastatic behavior of A3 files, which we were focusing in this research. Originally, we planned to use the actual curcumin and piperine extract from the turmeric root and black pepper, but the UPLC analysis results showed a lot of presence of the other components in the sample. So in order to prevent the contamination, we used the standards of curcumin and piperine for our cell treatments. So after performing such things, we found that the curcumin alone was showing the killing effects on A375, whether the piperine alone was showing the protective effects against the cells. That means like it was growing the cells a lot. But when we combined both of things with the tamoxifen, so like curcumin with tamoxifen and piperine with tamoxifen, it showed slightly different results, which means combining curcumin with tamoxifen showed that the cell viability was decreasing with the increasing concentration of the curcumin, but in the case of piperine, when the piperine was combined with the tamoxifen, the cell viability was increasing, such as at lower dose of piperine, the cells were growing higher compared to the higher dose of the piperine. So we found surprising result here that piperine can act as a protective agent against the tamoxifen. So when we use tamoxifen with piperine, that may not affect the cancer cells or help the patient to reduce the cancers. So to sum up, it can be said that piperine with the TAM had protective effect on cancer cells, whereas curcumin without TAM decreased cell proliferation. Additionally, adding curcumin to TAM did not increase in its effectiveness, but piperine reduced its TAM's activity and promoted cell proliferation. So here are the few references that I used to make the poster and that helped in my researches of curcumin and piperine and other methods. Last but not least, I would like to thank all my colleagues and my professors and lab technicians in order for constant support and guidance throughout the academic year and this research project. Thank you.